All right, welcome to our breakdown of a super important part of IB biology, Unit 3, Genetics. In the next few minutes, we're going to explore the genetic code that defines every living thing, including what makes you, well, you. Let's jump right in.
It's a pretty fundamental question, isn't it? I mean, everything from the color of your eyes to whether you're prone to certain conditions, the master blueprint for who you are is written in this incredibly complex language, hidden deep inside your cells.
So here's our roadmap for this explainer. We're going to start with the basics in the code of life, then look at how that code gets shuffled around and passed on. After that, we'll learn how to actually predict genetic outcomes, and we'll wrap things up by exploring the amazing field of biotech. Okay, let's kick things off with Section 1, the code of life. Before we can get into the really complex stuff, we've got to get a handle on the basic vocabulary of heredity. This is the foundation for everything else to come. So, right at the heart of it all is the gene. Just like it says here, it's a DNA sequence for a heritable trait. A really great way to think about it is like a single recipe in this massive cookbook that is your entire genome. You know, one recipe for eye color, another for making a specific protein, and so on.
But what happens if there's a typo in that recipe? Well, that's pretty much what a mutation is. It's a change in the DNA base sequence. Now, these changes can be totally harmless. Sometimes they're even beneficial, but in other cases, they can have a really significant impact on an organism.
And here's a really striking example of that impact. What you're seeing here is a normal disc-shaped red blood cell twisting into that crescent or sickle shape that's characteristic of sickle cell anemia. This drastic change in the cell structure, which can cause all sorts of problems like blocking blood vessels, comes from a mutation in just one single gene. It's a stark reminder of how one tiny change in the code can have massive biological consequences. All right, let's move on to our second section, genetic variation. It's not enough for this code to just exist. It has to be passed on. But it's not a simple copy and paste job. The way it gets passed down is actually what creates the incredible diversity we see all around us. The main engine driving all this variation is meiosis. That's the special type of cell division that creates sperm and egg cells. Now, meiosis has two key ways it shuffles the genetic deck: crossing over and independent assortment. We're going to focus on that first one, which is a huge source of genetic novelty. So that first method is called crossing over. As the definition explains, during prophase 1 of meiosis, homologous chromosomes pair up and literally swap genetic material. Picture this. Imagine you have two different colored strands of yarn, and you swap a few segments between them. You'd end up with two completely new hybrid strands. That's what's happening here. And this animation shows that process in action. See how the arms of the chromosomes overlap and then exchange sections? The result is what we call recombinant chromosomes. They're now carrying a unique mix of parental genes, creating brand new combinations that are a key driver of variation within a species. Okay, so we know genes exist, and we know they get shuffled up during meiosis. But can we actually predict the outcome of these genetic lotteries? Well, a lot of the time, yes, we can. Welcome to Section 3, Patterns of Inheritance, where genetics becomes a predictive science. One of the key tools for making these predictions is the Punnett square. It's basically a simple grid that helps us visualize all the possible genetic combinations from a cross between two parents, and it lets us calculate the probability of their offspring inheriting a specific trait. Let's use the ABO blood group system as an example. Take a look at this Punnett square. Across the top, we have the alleles from one parent. They're heterozygous for type A. And down the side, we have the alleles from the other parent. They're heterozygous for type B. Now let's fill in the boxes and see what the potential outcomes are for their children. And here are the results. Because the A and B alleles are codominant and they're both dominant over the O allele, that's the recessive I, this specific cross can produce offspring with all four blood types: type AB, type A, type B, and type O, each with a 25% chance. It's a perfect illustration of how recessive traits can pop back up in later generations. Okay, in our final section, we're moving from theory to practice, biotechnology. Our understanding of genetics has come so far that we're no longer just observing the code of life, we're actively reading it, using it, and even rewriting it. A classic example of this is how we produce human insulin for people with diabetes. As this flowchart shows, scientists can basically take the genetic recipe for insulin out of human cells, insert it into common bacteria like E. coli, and essentially turn these microbes into tiny living factories that pump out a limitless supply of human insulin. But how do we actually work with and compare DNA samples? Well, a key technique is called gel electrophoresis. This process separates DNA fragments based on their size, creating a distinct pattern of bands for each individual, almost like a unique DNA barcode. This is absolutely crucial for things like forensics, genetic screening, and, as you can see here, paternity tests. Okay, let's analyze this result together. Remember, a child inherits half of their DNA from their mother and the other half from their biological father. That means every single band in the child's lane has to match a band in either the mother's or the father's lane. So take a look at the bands that don't match the mom's. Which of the potential fathers has all the matching bands? If you look closely, you'll see the match is father 3. It's pretty incredible to think about how far we've come. I mean, in a relatively short amount of time, we've gone from just observing inheritance patterns in pea plants to sequencing the entire human genome, and now to developing tools that can edit that genome with precision. So we've journeyed from the basic code to how it's passed on, all the way to how we're applying that knowledge today. But this story is far from over. With revolutionary technologies like CRISPR, the ability to rewrite the code of life itself is becoming a reality, and that raises huge possibilities, and of course, some major ethical questions. Which leaves us with one final thought to ponder. What do you think the next chapter in the story of genetics will be?